Okay, so when we talk about the parts of the brain, here's a quick graphic of what we'll actually be looking at. The brain itself, many components, and different drugs will have different effects on different parts of the brain. So those neurotransmitters that we just went through, that long list, those are the ways that messages are passed along through the nervous system. And the whole process happens very quickly. So in order to allow those messages to be transmitted, the precursors for those neurotransmitters are found circulating in the blood supply. And the first step in the process is actually uptake. So this is when those precursors are taken up by the cells around them in order to create the neurotransmitters that the process will need. The next step will be synthesis, where those precursors are actually made into neurotransmitters through the actions of enzymes. So here's an image of how that would look. We have our precursor molecules and fragments, and then the enzyme that helps with the synthesis. They bind to the enzyme, and then the transmitter molecule is created. And here's an example of how that would actually look with dopamine. So our fourth step will be storage, where the neurotransmitters are stored in the vesicles. We saw that in the video on the neuromuscular junction. And then release. When that action potential arrives, that electrical signal traveling along the cells, then the neurotransmitters are released into the synapse, or that space between the nerve cells. And then binding, so the neurotransmitters are now onto the next neuron, attaching to the dendrites, and they can continue to have their effects after that. So we've got storage, release, and binding, where it crosses the synapse between the neurons, or between the neuron and the neuromuscular junction. And then metabolism. Once the signal's been sent, the neurotransmitters are no longer needed, so they may return to the neuron, or they may be metabolized, and there may be metabolites that are left in the bloodstream, and that's something that we'll talk about more later as well. Okay, let's look at a couple of examples of how drugs can become involved. We've gone through a lot of information on the nervous system, its components, and its functions. So let's talk about how drugs are actually related to this. So we went through our list of neurotransmitters, those important ones for cell function, everything from cell function to emotion. Now, drugs can actually alter whether those neurotransmitters are available in the synapse or not. We went through all those steps in the process of neurotransmission, and drugs can impact and affect all of those. So it might affect synthesis, where the neurotransmitter is being created. It might affect storage, release, and uptake, where it's crossing the synapse, or it might affect metabolism. An example of this, antidepressants. Many of these block the reuptake of neurotransmitters like dopamine or serotonin. And because it's blocking the reuptake, then it's actually increasing baseline dopamine and serotonin levels. And we suspect this is why those antidepressants are actually affecting mood in the people who are taking them. Okay, so there's one example, altering the availability of neurotransmitters. Another way that drugs can affect the nervous system is by direct action on the receptor. So the receptors, the dendrites, rather than receiving messages from the neurotransmitters, it may be receiving messages from the drug. So the drug is acting as if it was a neurotransmitter, and it's activating the receptors as that neurotransmitter would. Or the drug may actually be an antagonist, where it's occupying that neurotransmitter space and preventing it from activating the receptors that are key to it. So it could be taking the place of the drug, or it could be blocking the place of the drug. And these are just a couple of the ways that drugs can act on the nervous system. So because of all the interactions between drugs and its effects on moods, we've begun to wonder whether the the neurotransmitters themselves are what's really responsible for behavior. And this isn't a question that's new to the 21st century. It's not something that we're just trying to answer now that we have ways to scan the brain. We've been talking about these kinds of chemical theories of behavior for millennia, really. From ancient Chinese philosophy to ancient Greek medicine, we've attempted to explain people's behavior based on levels of chemicals in their body. A current one that's actually gaining some traction has to do with theories of mood. It's called the monoamine theory of mood. And we're talking about those same neurotransmitters again, dopamine, serotonin, norepinephrine. And we suspect that too little activity in these systems can cause depression, and too much can cause mania. But we're not able to explain all of behavior, even based on this particular theory, which again is it's been fairly well supported at this time. We haven't been able to come up with a single theory or a single kind of biochemical test we can run that really explains mood. And also, we're not able to find a similar conclusive theory or test for drug dependence. And even when we find things that seem like they might be important biological markers, we don't really see those holding up in an experimental setting. So we still definitely have a lot to learn in these particular areas. So just two more things that I want to touch on before we wrap up our lecture for today. We talked about brain imaging. As we continue to learn about the brain, we're learning more and more ways to basically get images of how the brain is working and what it's doing. Two important ones that we'll talk about this semester are PET, or positron emission tomography, and then the functional MRI. So a PET scan actually gives us a direct measure of brain activity. Now, to make it work, though, we actually have to inject radioactive materials into the brain. And we can see how the brain is acting at the time that the action is occurring. So some really interesting ways that we can start to learn about brain function and activity, but it doesn't tell us anything about brain structure while it's occurring. And then another tool that we're using is the functional MRI, functional magnetic resonance imaging. Now this one, we actually can see real-time changes in the blood flow in the brain, and it's not invasive, so we don't have that problem where we need to inject radioactive material. But this one, does not give us any information about brain anatomy while we're looking at it. So we're beginning to piece together the puzzle. We're learning more and more about the human brain here in the modern scientific era. We still don't have a single perfect tool, though, that gives us all the information that we'd like to see about the brain. Okay, so that brings us to the end of our material for today on pharmacology. The important concepts that we discussed today were the idea of homeostasis, the way that this affects the role of drugs and our experience with drugs in the body. We talked about some important structures in the brain, and then the action potential, the way that neurons communicate with each other, the various branches of the nervous system and the neurotransmitters that are associated with them. We also talked about neurotransmitters and the key brain structures and chemical pathways that they affect, and then, of course, the major functions of those neurotransmitters, the life cycle of the neurotransmitter molecule, and then finally some modern approaches to brain imaging. Be sure to be checking in on Web Campus for due dates and deliverables, and please reach out with any questions. Thank you, and have a great day.